Hello and welcome to the overview of SWAN. My name is Joe Delaney, creator of the SWAN software. This is a network-based pathway analysis. The data starts from gene-level data for each sample. Each gene and gene sample must have numeric data indicating negative loss or positive amplification. Networks are then drawn from some type of identifier. By default, this is protein-protein interactions from BioGrid. Networks are then further scored through phenotypes. The default is haploinsufficiency data, but users may upload whichever phenotype they prefer. This was originally designed for copy number alterations in cancer because previous software was not able to identify tumor suppressors and oncogenes within this very noisy data. The permutation analysis done by SWAN can deal with the noise and provide significant results. We've now tested this on a variety of different cases. This can be transcriptomic changes, or it can be single-cell RNA-seq changes. This is designed for biologists, not just bioinformaticians, but we do have software available on GitHub. The web-based software executes all 5,000 lines of code with just a few clicks. We have an emphasis on visual depictions of data within this web app because we know this is how basic scientists interpret big data. You have the ability to download the full tabular outputs and create your own plots if desired. And we also include example data to help get you started to know exactly what format is expected by the software. Data wranglers will help you get that data into a more SWAN amenable format. The overall workflow is to start with your data with samples in columns and genes in rows of a simple spreadsheet. This can be any type of numeric data. You may have controls or experimentals or just experimentals alone. You then input this into the data groomer, and this will help transform gene identifiers into gene symbols. It will also normalize data to be appropriate for SWAN. Once you have the data in the right format, you then perform a PAM pathway analysis. This is canonical pathway analysis. It helps you identify which pathways are most changed within your data and at what significance level. Once you know certain pathways which are most interesting to your biological question, you can then perform a more in-depth analysis using SWAN's single pathway analysis software. The data groomer can start with very esoteric names, which are just canonical identifiers from some output software, and then this is transformed into gene symbols, something that basic scientists normally refer to a gene as. And then it is normalized, centered around 0 with positive and negative data. SWAN pathway analysis can then be performed. This is an example of TCGA data. And then you will see an initial volcano plot presented. From that volcano plot, you can then infer which pathways are important to you. Then you can go into the single pathway analysis software. TCGA data is already uploaded if you want to play around with that, but you can also start with your own custom data. You then choose which pathway is important to you and which outputs you would most like from the software. SWAN is then run again on these samples, and a network is initially generated. This is the same network that is used in SWAN calculations and is a summary of all the samples within your data. It is interactive and you can push around the nodes as you like. The next most important plot is the feather plot. This is a summary of each individual gene within your pathway, and each gene is shown with each sample depicted on the y-axis. Each sample will have a particular shift associated with the data that you provided, and this gives you an idea of how the samples are distributed across each individual gene in the pathway. The comet shown in this gray line on top shows an overall cumulative sum of each gene within the pathway. Lastly, there's a Circos plot. This helps you know whether or not the network is drawn from genes on different chromosomes. This is available for human data and can help you identify exactly where your positive and negative hits are. Finally, SWAN has integrative analysis capabilities. You're not limited to just one type of data. You can upload up to 3 different types of data, such as copy number alterations, common data in cancer. You can then layer on RNA data, as well as mutation data, or any 3 data sets that you would like. SWAN will then perform an integrative analysis of all 3 of these data put together into one analysis. So with that, I hope you enjoy our analysis software and find it very easy to use and informative for your own data sets.